So a very famous nitrogen ring structure is that of heme. So you probably heard of hemoglobin. It's the main protein responsible for carrying oxygen around the blood and to cells within the body, which will be very important, as we'll explain in a little bit. So what you might not know is that hemoglobin actually has this component called heme bound to it. And if we look at the structure of heme here, it's actually got 4 of these nitrogen rings that can be considered amide containing rings. So there's 4 rings here, and there's also a connection between each of these rings via the carbon atom. So there'd be a carbon atom in these positions that I'm drawing. So we've actually got 4 interconnected rings that have the nitrogen there. So there's actually 4 amide groups. So what's really important about heme is that it's able to basically hold an iron atom in the center here. So we can see it represented by its chemical symbol, Fe. So we've got this iron atom in the center of our heme group. And this is extremely important because this is the position where oxygen will actually come in and bind. So oxygen is held in place by the iron atom within the heme group. But without the heme group, there's no iron atom there to hold the oxygen. So heme plays a central role in being able to bind oxygen. So as I said, we have heme binding to the hemoglobin protein, which is present within our red blood cells. So let's have a look at this relationship in a little bit more detail and see how it's actually really important. So hemoglobin is the protein that is thought of as the oxygen transporter within the blood. So the oxygen will actually bind to the Fe atom at the center of the heme ring, and that heme ring is actually bound to the hemoglobin protein. So essentially, you can't have the heme ring functioning without being bound to the hemoglobin protein, and you can't have oxygen binding without the Fe being held at the center of this heme ring. So they're all very dependent on each other to function. So essentially, you need the hemoglobin protein, you need the heme ring, and the Fe atom in the center. And if you've got all those 3 things, then oxygen can bind to the Fe atom and be transported around the blood within the red blood cells and be delivered to where it's required. So once the oxygen is bound to the hemoglobin within this heme ring here, then it can be transported. So essentially, we take the oxygen from our lungs via the blood to all the different tissues and organs to make sure that the cells within these tissues and organs have enough energy in the form of ATP to continually perform the processes required by the cell and maintain the structures of the cell. So if your cells are starved of ATP for any length of time, they'll actually start to die. And that's actually what happens in gangrene. So if you've ever seen pictures of gangrene, it's usually when like a foot or a limb goes very black, and that's essentially the tissue dying due to oxygen supply being cut off. And that corresponds to many cells, huge numbers of cells, having been cut off from oxygen and not able to sustain themselves properly. So essentially, if the body can't get oxygen to the cells, the body dies. So this is why you'll suffocate without a supply of oxygen. The body will just shut down because it can't make enough energy to keep all the systems going. So let's have a little bit more of a graphical representation look at how we're actually able to carry the oxygen and how these structures relate to each other. So hopefully you know that within the red blood cell is where we're going to be carrying the oxygen. So we've got the red blood cell here represented as the red circle, and within the red blood cells there's going to be a very large number of these hemoglobin proteins, which are the oxygen carrying proteins. So if we zoom in on our red blood cell, we can now see these hemoglobin proteins, which are represented as these green spheres. So Obviously there's going to be a lot more than 3 of these, but just for simplicity I've shown you that there's multiple of these hemoglobin proteins within the red blood cells. So if we zoom in even more, now we've got a view of one hemoglobin protein. And within a hemoglobin protein, we have a heme ring attached. So the heme is a compound that will attach to a specific position within hemoglobin. And because the heme ring holds that Fe in the center, it can actually bind to oxygen. And you can see that represented with the O2 bound at the middle, in the middle of the heme ring here. So the oxygen molecule is bound to the heme ring inside the hemoglobin protein. So essentially we're reliant on all 3 of these structures to ensure that oxygen can be transported around the body. We need the red blood cells to carry its contents. We need the hemoglobin protein within those red blood cells to bind to the heme, and we need the heme to bind to the oxygen. So if any one of these pieces was missing, the whole process wouldn't work. If you took away the red blood cell, there'd be no hemoglobin or heme. If you took away the heme, there'd be no oxygen binding capacity of hemoglobin. So all of these structures are absolutely essential to oxygen transport. So here's a summary of some of the other biologically important amines. So if you look at each of the structures here for dopamine, adrenaline, and histamine, the commonality is that they all contain a nitrogen atom, which is part of an amine functional group. And in the case of histamine, it's actually got 3 separate amine functional groups. So if we look at other parts of the compound, there's actually other functional groups present as well. Histamine doesn't have any other functional groups, but what about dopamine? Can you recognize any other functional group here? Well, let's say this part here is one functional group. So we've got a benzene ring and an OH. So what do we call that functional group there? So when we have the OH and the benzene ring, that's a phenol functional group. And coming off that phenol group is another OH group. So that is alcohol. So you may be wondering why I didn't say that this OH group here was part of the phenol ring. It's arbitrary. So you could say that either part was a part of the phenol ring and the other OH is the alcohol group. Similarly, in adrenaline, we've got this same arrangement of the phenol and the extra OH. And we've also got an additional OH down the bottom here, being another alcohol functional group. So let's have a bit of a look at some of the properties of these important biological amines. So dopamine is actually a neurotransmitter, and it's a stimulant that's produced in the brain, and it actually provides feelings of enjoyment. So there's a couple of ways that the dopamine can actually be interfered with. So for example, in Parkinson's disease, one of the consequences here is actually a deficiency in dopamine. So that's part of what's happening in Parkinson's disease. Also, through the use of cocaine or other amphetamines, basically what these do is actually inhibit dopamine being moved on, and it actually stays longer in the synapse between 2 nerve cells. And basically this means that the feelings of enjoyment that are being generated are much longer lived. So this is the reason why people choose to do these types of drugs, because of the feelings of enjoyment you get. So it's manipulating this neurotransmitter here. So adrenaline, I'm sure we all know that it's part of the fight or flight response. So essentially the hormone is released when you're faced with a situation where you either need to stay and fight or flee. And adrenaline has many functions, including activating some metabolic processes to give you quick energy, but it can actually function as both a hormone and a neurotransmitter, so it's quite diverse. Histamine is an interesting compound as well. So some of you, particularly if you're allergic to things, would know about histamine. So it's actually what we call a pro-inflammatory mediator. So it induces inflammation, and it often is in response to allergic reactions. So when your cells come in contact with a particular allergen, it releases this histamine compound as a way of starting the inflammation process. So one thing we've come up with to try and lessen the effect of these allergic reactions is actually to use antihistamine drugs. And what they actually do is they'll actually bind to the receptors on specific cell types that would normally bind to histamine. So it's kind of like blocking the ability for histamine to signal. So if we've got a cell here that has a receptor that can normally bind to histamine, so here's our little histamine that can come in and bind here. So once you have a binding event between the histamine and the cell, then you get the inflammation process being basically switched on. So this will happen to lots and lots of different cells, but just showing you one as an example. But what about if we bind something else to that receptor there? So we're going to bind one of these antihistamines. So if we bind an inhibitor or antihistamine into that site, basically what that means is, even if there is histamine floating around, it actually can't come in and bind here because there's no space for it. So essentially what the antihistamine does is it just blocks the position that the histamine can normally bind to the cell, which we call a receptor. Alright, so now we've reached a participation plus question. So remember, these questions are really handy for confirming your understanding of what we've done so far in the session and see how different things link together. So I encourage you to have a go at this question and also share your answers with your peers and get some other perspectives, because it's handy to hear about how other people approach these questions to get a different perspective. So we've also reached the stage in the session where you can have a go at revision questions 3 to 6. So as always, you can access these through the revision quiz on the LMS. So I encourage you to pause the presentation and have a go at these now to try and confirm your understanding of what we've been through thus far. So to summarize what we've seen in part 2 here, which is the amine part of the session, we saw that nitrogen had a specific way of forming covalent bonds in terms of needing to pair its 3 unpaired valence electrons, which meant it could form 3 covalent bonds. To form these covalent bonds, it could either form 3 single bonds, a double bond and a single bond, or a triple bond. So by having 3 bonds, that meant the nitrogen atom had satisfied the octet rule and had 8 valence electrons, which made it very stable. So we then looked at the amine functional group, and we saw that it was where we had a nitrogen atom connected to 1, 2, or 3 R groups. And I also showed you that we could specify between the 1, 2, and 3 R group amines by saying whether they were primary, secondary, or tertiary. So if it had 1 R group, that corresponded to 1 attachment to carbon atom, and that made it primary. If there were 2 R groups, it corresponded to 2 carbon attachments, and if there were 3 R groups, then we had 3 carbons attaching to the nitrogen and giving us that tertiary amine. We then looked at some of the properties of the amines. So they have a smell resembling raw fish. In particular, we looked at trimethylamine, that was a very fishy smelling amine. But interestingly, we can convert these smelly amines into odorless amine salts by undergoing an acid-base reaction to turn the amine into an amine salt. Creating these amine salts is also useful for amine-containing drugs to make them more soluble in water and also to give them a longer shelf life. We also had a look at our cyclic amines, which is where we have the nitrogen within a ring structure. So we've got the nitrogen atom bonded to 2 different carbon atoms, which are part of the same ring. So some quite famous cyclic amines include the nitrogen bases within DNA and RNA, and also the stimulants like caffeine and nicotine. Spent a bit of time also looking at heme, which contained 4 of these cyclic amines. It actually binds to the hemoglobin protein within red blood cells, and it is the heme ring that coordinates the Fe atom which is responsible for binding oxygen and transporting that around the body, which is vital to enable our cells to get enough oxygen to make the energy they need to keep everything going and survive.